All right, welcome everyone to this panel discussion, continuing the Genomic Prediction Webinar Series. I'm Lawrence Tellier, CEO of Genomic Prediction, and I'll be the moderator today. We have a fantastic subject, the results from the recently published Nature Scientific Reports paper titled Sibling Validation of Polygenic Risk Scores and Complex Trait Prediction. So let me introduce our panelists. First, Professor Simon Fishel, early IVF pioneer, is president and founder of the Care Fertility Group. Professor Fishel has worked 10 years with the Nobel laureate for IVF, Professor Sir Robert Edwards, from 1975 to 1985, and was part of the original team of 4 with Patrick Steptoe and Job Webster, who opened the world first IVF unit, Bourne Hall, outside of Cambridge in 1980. Professor Fishel is known for his long history at the forefront of every major technological breakthrough in the field of IVF, including this one. I'm also particularly humbled to announce today the founding of Genomic Prediction Scientific Advisory Board. We are deeply honored to appoint Professor Fishel as the head of Genomic Prediction Scientific Advisory Board. Dr. Jamie Metzel is a leading technology futurist and geopolitical expert, a faculty member of Singularity University Exponential Medicine, and founder of the global social movement, One Shared World. In 2019, he was appointed to the World Health Organization Expert Advisory Committee on Human Genome Editing. He's the author of 5 books, including the recent bestseller, Hacking Darwin, Genetic Engineering and the Future of Humanity. Professor Steve Hsu is a founder of Genomic Prediction, is also a professor of theoretical physics and professor of computational mathematics, science, and engineering at Michigan State University. Professor Hsu has also been a force behind much of the science underlying genomic prediction as the senior author of the paper we'll discuss today. Steve, the floor is yours.
All right, thank you very much. Well, gentlemen, I am sharing my screen with you. I hope you can see it. Now, because I'm sharing my screen, I can't see your lovely faces. So if you raise your hand to get my attention, I won't be able to see it. So just you'll have to use your voice or something like that to get my attention. Our goal today is to talk about the current status of polygenic risk prediction, and in particular to discuss the results in this paper that was recently published in Nature Scientific Reports. And what is unique about this paper is that it uses tens of thousands of sibling pairs. So these are people who are already late in life, maybe 60 years old, and whose health history is known and whose genotypes we know. And because they're already late in life, we sort of know what health conditions, what diseases they've experienced and what they have not experienced. And it's a very fertile ground, a kind of ideal setup in order to test whether polygenic risk prediction works in differentiating between siblings who have actually been raised in the same family and experience the same environment growing up. So it removes a bunch of the standard confounds of environment versus genetic causation. And now this is a kind of, just based on what I already told you, it sounds already kind of like it could be a technical subject. The goal here, though, is not to make this complicated. The goal is to try to convey this in as simple a way possible for non-experts, for people who are not experts in computational genomics or polygenic risk prediction. So I encourage you guys to ask me questions. Think of the audience, so ask if anything I say is unclear or you think might be confusing to a layperson who's nevertheless interested in this subject. Please feel free just to ask. And what I'll do is for each slide, there are just I think 6 or 7 slides here, and for each slide what I'm going to do is just talk a little bit about the content of the slide, and then you should feel free to interrupt me or at least ask a question once I've completed that initial description of the slide. You don't have to wait till the end of all the slides to ask questions. So let's get started. This is the paper. You don't have to read that abstract. The font is probably way too small for anybody to read it, but if you follow that link on the lower left, you can find the article. And we've deliberately published a lot of our papers in Scientific Reports, which is the open access part of Nature. So normally to read Nature articles, you need a subscription to Nature, which is quite expensive, and typically only people at academic institutions have such a subscription. So we prefer to actually publish our results in the open access part of Nature, which means you can just go to that link and read the article even if you have no Nature subscription. So the background for all of this science, which has really progressed very rapidly just in the last few years, is the ability to inexpensively read out the genome of an individual. And so you may remember from school that there are about 3 billion base pairs in your genome, and that two random individuals, say you and I, will differ at roughly about 1 per 1,000 locations. So if I take 1,000 locations, individual base pairs, you and I will be the same at almost all of them, and there'll just be maybe 1 out of 1,000 differences. So if you take 1 out of 1,000 out of 3 billion base pairs, that's typically a few million differences between any two people on average. And what scientists have figured out is the places in the genome where people are most likely to differ from each other, where there is common variation, commonly found variation between individuals. And there is an inexpensive technique called a technology called a gene array, which specifically tries to read out the millions of locations where individuals are most likely to differ from each other. And that tends to capture most of the common differences between people that's encoded in their genome. And then what happens is when we assemble large data sets, so data sets with hundreds of thousands, maybe even 1 million people for whom we have their genotype, and then we have some information about them. So for example, whether they've ever had diabetes or whether they've had breast cancer or how tall they are. Those are called the phenotypes. And using methods from AI or machine learning, we have to train a computer to look at the DNA of an individual and then make some prediction about that individual. And the main topic of our discussion today is the validation of those predictors, measuring the goodness of those predictors or the quality of those predictors, and specifically when tested on pairs in which the two people in the pair are siblings, say two brothers that grew up together. So let me stop there. Are there any questions on this first slide?
So how close are the siblings compared to you and I? So the information.
So yeah. So I think I understood the question. So instead of this 1 per 1,000 differences, there are roughly half as many differences between the siblings because they sort of the DNA is scrambled up from the mother and father, but the brother and sister are more likely to be similar on any particular base pair than say you and I would be.
And so the information published on this incredible cohort that you've got is obviously going to be tighter and more informative, I presume.
Correct. So actually, it's more challenging to make predictions between siblings from the genetic level because they tend to be a bit more similar. So we're actually working on fewer differences in each pair than we would have if you just took two random people, like say you and Jamie. So it's challenging in that way as well. And I think most experts would say that if you can do good prediction between siblings, that is really the gold standard to be sure that the predictor is actually capturing some kind of causal genetic effects.
Thank you.
So Steve, the genetic information is inherently standardized, but the phenotypic information is not. How do you think about standardizing the different types of phenotypic information that you have in these data sets?
That's a great question. So in the current state of the art, when we're doing disease conditions, we actually have a very crude phenotype. It's basically 0 or 1. So it's take a person who's already in their 60s and ask, do you suffer from diabetes or not? Do you suffer from heart disease or not? And rather than having a more nuanced measure like the severity of the condition, generally what is recorded in the medical records that we have access to is just, do they have this condition or do they not? For quantitative traits, like say the height of the individual, we typically have that down to about centimeter accuracy. But for disease conditions, we're just basically training on data which knows which individuals late in life have suffered the disease or suffer the disease, and which ones have not.
And could I ask, presumably with the phenotype, that would also have there would be an environmental effect on how they've managed it themselves. So the phenotype is actually much more imprecise.
Yes, that's correct. And I just want to emphasize to everybody listening a very basic point, which is that we're not claiming that your DNA is your destiny. We're talking about predispositions. So something about your genetic makeup that makes it more likely that you might suffer from diabetes. But of course, what you eat, whether you exercise, other environmental aspects of your life will also affect your disease status. Okay, let me go on to the next slide. You guys are great. These are great questions, but Laurent might yell at us if we go too long. So let me go to the next slide. So these are two different disease conditions. So breast cancer in the left graph, and coronary artery disease for men on the right. And these are two conditions for which we have pretty good predictors. And I just want to spend a little bit of time talking about them. So now already these graphs are a little bit complicated. Let me focus on the breast cancer one just to take it as an example. So the red curve is describing women who have been evaluated based on their DNA alone to be high risk. So what it means is that when we score their genome, they are ranked in the top 3% among all women for being high risk. The green line describes women who, when we evaluate their genome, the algorithm rates them as the lowest 3% risk in the population. So they're the least likely to have breast cancer. And the blue line is taken from sort of the middle of the bell curve. So it's women who are between 40th and 60th percentile risk. So it's basically the average sort of typical woman. The horizontal axis on the graph, I guess you can see this mouse here. The horizontal axis is their age in years. So we have their medical history at various ages. And so we actually know if any of them received, say, a breast cancer diagnosis, we know at what age it happened. The vertical axis here is the percentage of a particular group which has been diagnosed with breast cancer. Okay, so let me tell you a story. For all the women, regardless of what their PRS score was at the beginning, when they're young, it's very unlikely that any of them have breast cancer. As they get older, you start to see dramatic differences. The women that were calculated, again, from DNA alone, no other information about the woman, just the DNA, the ones who are predicted by DNA to be high risk start being diagnosed with breast cancer at a much higher rate than the average, the sort of typical middle of the bell curve women, and much, much higher than the low risk women. And so what these three lines tell you is the life story of women that in a sense could have been foretold when, say, maybe they were first embryos from the DNA alone. A subset of them were much more likely to have breast cancer than another subset. And I could tell a similar story for the right graph for men. And in this case, the condition is coronary artery disease, not breast cancer. But you'll agree these are sort of heavily impactful, common disease conditions that really are affecting millions of people around the world. Now, I want to make a special point about breast cancer. So many of you are probably aware of something called BRCA. So there are rare genetic mutations called BRCA mutations, which also increase risk for breast cancer. We are not talking about BRCA here. So when we classify the women on the leftmost graph, we are not using BRCA, and all of these women are negative for BRCA. They do not have the rare mutation, okay? What's interesting is that only a few per 1,000 women in the general population have a BRCA variant. So only a few per 1,000, maybe 1 per 400, will have the rare mutation that predisposes. Now, what's interesting is that if I ask how many women are at high risk for breast cancer due to BRCA, which is well known, versus women who are at similar risk due to polygenic risk, which is a new thing, which could not have been diagnosed or figured out just a few years ago, but now can be done fairly low, fairly inexpensively. It turns out there are about 10 times as many high-risk women due to polygenic risk. So these are women we can identify today, but could not have been identified just a few years ago, as there are women who are at high risk due to BRCA. Okay, so if you want, to the extent that BRCA made a big impact on the way breast cancer is dealt with in medicine, so women who have BRCA variants tend to be asked to take mammograms and extra screening earlier in life than other women. A much larger population of women now can be treated in that same way, and hopefully we will detect and be able to treat their breast cancer earlier in life thanks to this kind of genetic technology. Now, one interesting aspect of this is that if you have a family history, imagine a family where if you look at all the aunts in the family, grandmother, great-grandmother, maybe they have a preponderant, maybe they have a family history of breast cancer. And so the family as a whole are sort of high risk for some genetic reason. Well, it turns out it's 10 times more likely that that family is high risk due to polygenic effects than to monogenic or BRCA effects. So what we'll actually talk about in our research when we looked at all these siblings is we found many families where, because of a family history, they had higher average polygenic scores, higher average polygenic risk than the general population. And so now we can understand what is going on in these families. So these are families that were BRCA negative, so there's no reason to think that they had a preponderance of breast cancer because of BRCA. There must have been some other genetically inherited cause, and now we start to know what it is. So let me stop there. The purpose of the slide was to just give you an overview of where the technology is today for polygenic risk scoring, and to give you sort of two very impactful examples.
So Steve, I'm imagining that when you play this forward 5 or 10 years, what we would see is that every person would have a health score that would incorporate their polygenic risk with their other lifestyle risks, other biometric information, family information, and all of that would create a comprehensive risk score that would be the most actionable item within the context of healthcare. Is that how you're thinking about it?
Yes, I think if we were to think about how this will affect adult health going forward, say a decade from now, hopefully sooner, because the science is very strong. It's just a matter of getting it out there in the health systems. You can imagine you go to the doctor, they take a saliva or blood sample, and relatively inexpensively, they measure your genotype, and then we compute all of these scores for you. And there are now about 30 disease conditions for which we can compute scores like the ones that you're looking at here. And for most of those scores, you're probably in the blue category. You're probably not super high risk and not super low risk. But out of those 30 disease conditions, and in 5 or 10 years, it'll probably be 50 disease conditions or 100 disease conditions, some of those disease conditions, you'll be exceptionally low risk. So maybe you don't need to have a colonoscopy, okay, right away. But on others, you'll be super high risk. And for that handful for which you're high risk, that is what your general practitioner or personal physician will be especially on the lookout for you. And so it's sort of ushering in the beginning of what we might call precision medicine based on genomics. It's a weird accident of history that these kinds of scores are available to parents going through IVF for each of their embryos. It's available to them now for their embryos before it's generally available in the health system for adults. And that's just a weird accident of history.
But it's extraordinarily powerful. And it's as you mentioned, it's the ushering in of this partial holy grail in medical practice, which is personalized medicine. And it seems to me that anything we can do to tailor treatments more specifically, and of course you will presumably learn the phenotypes a lot more in relation to this DNA data, as to whether or not, for example, one undertakes prophylactic bilateral mastectomy, for example, because this is you know, it goes back to what it used to be like when they used to do surgery because it was the only thing that was available to them. It seems a very powerful option, as Jamie says, for adults or newborns, just as much as it is for embryos. It's just this accident that it's come available for embryos. And as you say, Steve, this seems to be just one example of the power of this technology.
Yes. I'm going to show you more examples as we go on, but yes, and one thing I want to emphasize, I guess I'll show this on another slide, but it's good to emphasize it. You know, in making this figure, they chose top 3% and bottom 3% as just an arbitrary grouping, just to show the difference between high and low risk. But we can go further in the tail. We can look at the top 1% or top 0.5%, and you'll see curves that shoot up even much more strongly than this for those individuals. And so think how powerful it would be if you just happen to, on 100 different disease risk predictions, there's one of them that you're just really a very strong outlier. And we can say, sir, I'm sorry, but we think there's a 90% chance you're gonna have hypertension when you're old. You know, that's almost like science fiction, but for some subset of cases that will be true. Someone will be so far in the tail that you'll be able to tell them with sort of better than 50-50 or maybe 80-90% accuracy that they're going to have the condition when they're old.
And I know you're going to come on to this, Steve. So I just want to emphasize then what this is suggesting to me as well is that the earlier this information is available to any person, especially if they're at an embryo stage, as it were, the better, because in the end, we will gain more and more knowledge on clinical management of this DNA information.
I totally agree. I think even just 10 years from now, I believe young doctors will say, it was barbaric. You know, there were people going through their lives without knowing that they had unusual risk for these 5 conditions and almost no risk for these other 5 conditions, and the rest they were kind of in the middle. And it's barbaric. People went through their whole lives and didn't know this. Now we immediately diagnose this at the embryo level or at when the kid is born. Mm-hmm. So I think we're just 10 years from that probably. Mm-hmm. So let me go on to the next slide. So here are 2 examples, and these are slightly different kinds of graphs. So rather than plot the progression in time as somebody ages, what we have here is the percentile of someone on the PRS score, polygenic risk score. 1 meaning you're at the high end for risk, let's say 99th percentile, 1 percentile, 2 percentile meaning you're at the low end. And the green line represents the typical person. So the green line is sort of the average risk that someone in the population has. The red line is the theoretical prediction. So based on the algorithm, based on the risk score algorithm and what we know about it, we can make a theoretical prediction of, conditional on where you are in your risk score, what is the absolute probability that you'll have the condition? So say 20% or 30%, et cetera. And the blue dots are a test. So this is what we mean by validating the predictor. We go to a completely different population than the one that was used for the training of the AI predictor. We go to a different population, and then we just ask people in the we genotype the people in the population. We say, and then we find out, did you have breast cancer? Did you have hypothyroidism? And within a particular group of people, say we bin together all the women who are at the 82nd percentile in the validation set for risk for the PRS score, and then we ask what fraction of those women actually turn out to have been diagnosed with breast cancer, and that is the blue dot. So the blue dots are real validation data, and what you see is they hug that red curve pretty well. Now what's not shown here is the error bar. So sometimes if in a certain bin there just aren't a lot of people, we plot the dot, we put the dot where the fraction of the people is who have the condition, but there's a big uncertainty in the derived probability because there just weren't that many people in that particular bin. So the noisiness, the scatter in these blue dots is because just statistical noise of not having enough people to really pin it down. In the case of hypothyroidism, we had more data, and so you can see that the blue dots are hugging the theoretical curve very tightly just because there's less spread. In each of the bins, we have a large number of people, and so we're able to estimate the absolute probability pretty well in each bin, even in the tails. And the thing I want to emphasize is how this red line shoots up as you get to 199.9% risk percentile, and how it shoots down for the lucky people who are at super low risk for the condition. And we see this, we've validated this, and other researchers have validated this for many different, again, many different disease conditions, about 30 different disease conditions now. So the general takeaway is that if you're in the middle here, your polygenic score is not telling you very much. There's just not that much difference between being here on the curve and being here on the curve, because it's kind of flat. It's almost horizontal. It's almost like that green line. But if you're one of the people who's in the far left tail or the far right tail, then it really speaks to you. And so similarly with embryos, most of the embryos are on a particular predictor going to be kind of typical, and there just isn't that much to say. But there are going to be some embryos where we can say this embryo really has very low chance of hypothyroidism in life. This embryo looks like 75% chance of hypothyroidism later in life. And so it's really at the tails of the distribution, the endpoints, where you can really make strong statements. In the middle, you can sort of, it's best probably not to make very much of a statement and just tell the patient that, we don't know very much about how you're different from an average person if your score is somewhere in the middle here. So let me stop there and answer any questions you have.
So presumably as the data sets get bigger, the granularity with which we'll be able to look will increase. So I know you use the UK Biobank. I mean, it seems like there would be other resources that could be used, like blood banks or other places. We've talked about veterans, where we could really build up a much bigger data set. What kind of numbers do you feel we'll need in terms of total data sets to bring this to a new level of accuracy?
So we're going to see steady, constant improvements in the quality of these predictors, and we have just in the last few years. The UK Biobank is about 500,000 people. In the UK, which is a real leader in this kind of science, they are planning to recruit 5 million people, so 10 times as many people, through the NHS, the National Healthcare System. So they're going to generate, probably within the next 5 years, a 10x larger data set than we currently have available. My group also works with, for example, the Taiwan Biobank, which has hundreds of thousands of individuals with medical records. There's a Japan Biobank, which is of similar size. And so it's really just a matter of working. Like when I meet with my team, we talk about, okay, do we have access to the do we have access to this particular autism data set? No. What do we need to do to get it? And then what calculations are we going to do once we get access? So there's no shortage of work to be done in this field.
And Steve, given that in some diseases there are clearly ethnic differences, what kind of computations do you need to make, or any at all, with my very limited understanding of this, would you have to make for ethnic variances?
So that is an unsolved problem. That's one of the main problems my lab and other labs are working on, is that if you train your algorithm on, say, people primarily of European heritage, what modifications do you need to make to the predictor in order that it works well, say, on people from Japan? And it's really an open question that we're working on. Right now, the main method that people would use is they would try to find a population of, say, Japanese people in order to build the predictor for Japanese people. But as we get more of these predictors and we start to look inside them, we start to learn how we could have maybe constructed that Japanese predictor without a large sample of Japanese by just modifying the European predictor and knowing something about the difference in the genetic distributions of those two populations. But the short answer to your question is it's actually an open question under very serious research right now.
And so,
okay, let me go to the next slide.
Yeah, please carry on. Please carry on. No, please, Ken.
Okay. Okay, so now we're, I think, if I can make this advance into my slide. Oh, this was, okay, here we go. Sorry, Lauren, you'll have to edit this out. Something just froze in my.
No problem.
Okay.
Fortunate submarine.
Yep.
So now we get to the paper, the specific paper that we're talking about. And here what we're doing is an analysis where we specifically focus on pairs of people. And there are two kinds of pairs we might form. We might form random pairs where the two individuals in the pair are not really related to each other, not in any known way, or they're closely related. They're actually siblings who grew up together. And we want to compare our ability to pick out which of the two individuals in the pair has the condition based only on the DNA, based only on the PRS score. And you can see that when we're talking about siblings, this is exactly analogous to the embryo selection problem. So in other words, you're trying to make the best choice for the family, and you're choosing literally between siblings because those embryos are siblings. And the question is, how well does the prediction perform? And we can compare it as a benchmark to how well it performs when you're just trying to pick between random people. And so that really is the main focus of the paper. And of course, we've got some confusing, weird graphs that only geeks love, which I will now walk you through. Okay, so let's take, for example, this one, hypertension. The red line here in all four of these graphs is always for random pairs. So these are people who are not related to each other as far as we can tell, and we can sort of tell from their genome that they're definitely, even if they didn't know they were related to each other, we can tell for their genomes they're not related to each other. The blue line is siblings, pair of brothers, pair of sisters. And what we're comparing here is on the horizontal axis, we're talking about an increasing risk score. And we're asking, what is the probability, given that one of the twins is at high risk? So one of the pair is at high risk. So the number on the horizontal axis is describing the score. It's in something called standard deviation units. It's the score of the high risk individual in the pair. And then the vertical axis is telling us how often that high risk person is actually the one that has the condition. And you can see we can get to prediction probabilities of something like 80% for some of these predictors, 90% for some of these predictors, which means that when we have one sibling, for example, or one individual from the population who's sort of normal risk, and then we have another one who's high risk, our ability to predict that the high risk one is actually the one who has the condition can be very strong. It can be 90%. And so it turns out the accuracy, if you just look by eye, of the red line is not really very different from the accuracy of the blue line. The fatness of the shading around the line is the statistical uncertainty. And it can be fatter if we just don't have that many examples. So it's always fatter in the blue case because we have fewer siblings to work with than we do just random pairs of individuals because there's just more data of that type. And so the fatness of it is just giving you some estimate of the uncertainty that we can really just estimate the probability. But the fact that they overlap so much is telling you that in layman's terms, basically picking between siblings works about as well as picking between random individuals from the population. And furthermore, the probability that we call correctly which of the individuals in the pair has the condition can be very high. It can be 90%. So let me stop and answer questions.
So Steve, is this for all 30 or this is, oh, I see. So this is one for each. So if you're picking between embryos and let's say you had 30 different disease states that you were picking, then you would add them all up. And I imagine in the best case scenario is nobody is an outlier on a bad thing, but in some scenarios, there'll be a small number of embryos that will be outliers with potential negative outcomes. Is that right?
Right. I think the most the clearest and simplest use case is imagine you have a family history of type 2 diabetes. So that would be the upper right panel, this one, right? So you have family history of type 2 diabetes and you have a few embryos to pick from, and one of the embryos is an outlier, has high risk. you can be confident that the high-risk one really is at significantly higher probability of later in life having type 2 diabetes than the other two. That's basically what these results are saying.
So you could imagine a number of scenarios that's being faced by the patient and by the clinician, and clearly there's going to be a lot more training of clinicians going on here too as well. But I think that's fairly easy to deal with because this is so important. But you will have variations that they will have to make choices on, or you could actually find that they're all a pretty similar risk, and why did I bother checking in the first place? And the answer to that would be, well, it's just like when we want to check for chromosome abnormalities, you might be lucky and they're all normal, or you might be actually unlucky and they're all abnormal, which was worth doing. So I think the point is here is the power of being able to have choice, even if the choice is an easy one, because actually it may not be easy at all, but at least you have choice, which we've never had before. And the choice isn't just about which embryo to transfer. It's about looking after that human being for the rest of its life post-birth.
Yeah, I think that was a great summary, Simon. I think one of the things you touched on, which is for aneuploidy, so looking for abnormalities in the chromosome structure, we're used to the idea that, it's only 1% of embryos that will have a problem. I just took that number, but let's say 1% of the time you have a problem with trisomy or something. But the impact of it is so strong that you actually are willing to check, even though most likely the answer you're going to get back is, oh, the embryo is fine. You didn't need to do this check. It was fine. But because the impact of this is quite strong, you care, and it's worth doing. So similarly, if some embryo is top 1% for breast cancer or for coronary artery disease, we saw that in the previous graphs that there was a pretty material difference in the way that their health might go later in life. And so, again, most of the answers you get back on a particular embryo are going to be, it's in the normal range for coronary artery disease. It's in the normal range for type 2 diabetes. It's in the normal range for hypertension. Oops, for hypothyroidism, it is an outlier. And we just wanted you to know that. So I think that's going to be the flavor of these reports. And it is more complicated for the physician to figure out, but I think physicians are smart. And, I think after a little bit of training, they can understand all of this and they can bring a lot of value to their patients.
Yeah, and then I think it's also going to require a lot of training in just counseling and interpreting the data. Because, so let's say we have these 30 disease conditions and it goes up to 40 and then 50, but that's 40 or 30, 40 or 50 out of a much larger number. And so when people think of a baby, they don't think, oh, it's hypertension avoidance is the activity of making a baby. And so it's because we all have this model of perfection in our minds. And then you see something which is a chart, and there's one indicator that's a probabilistic concern. It's going to require a lot of sensitivity to place that within the proper context.
Absolutely. And this isn't the topic of our current discussion, but one of the things we've been working on at the startup is something called a life index, which helps the physician to aggregate all the information that's in these, say, 30 different predictions about different embryos. And how do you aggregate all of that maybe into a predicted sort of impact on quality of life or impact on life expectancy, et cetera. Some aggregate metric, I think, will make life much easier for the physician and just kind of picking between embryos.
Well, I think that's going to be essential, Steve, and that's not just for the physician, but also for the patient to understand, because we need to get to a point where, at the moment, if people have a concern over this technology, they much prefer to roll the reproductive dice. That's fine. That's up to them. But many people would want to have the option to have information, and so this choice that they've got is really important. But it needs to be laid out to them in a way that they can understand what it means post birth. I mean, that's what this is all about. This is when they get the baby. What does this really mean? And it's that aggregate metric when it's eventually understood in a format that can be very rapidly delivered from the outcome of the embryo as a choice, I think that's going to be critical.
I agree. We don't want to paralyze people with choices that are too complex, and so you know, some middle ground has to be found where there's some kind of aggregate measure of you know, overall health for the embryo. Okay, let me go on to the next slide here. These are just two more examples, so let me just go through quickly. This is type 1 diabetes. Again, you can see that when one of the two is high risk, we generally 80 or 90% of the time are picking that. It is the high risk one that ends up having diabetes, type 1 diabetes, relative to the normal risk one. And the standing height result, this is a different thing. This is it's not something we actually offer, but it's just to show the quality of that predictor. In words, what I would say, what this graph tells us is that if conditional on there being, say, at least a 2-inch difference, so two brothers and one of the brothers is 2 inches or more taller than the other brother, we call the taller brother correctly. The algorithm calls the taller brother correctly over 80% of the time. And once there is a more significant difference, 2 inches among brothers is really not that great, but if there's a more significant difference, like say 4 inches or 5 inches, then we can reach 90 or 95% accuracy in picking out which one is actually taller. So, you know, there's a level of accuracy here that is going to be reached, I think, across many different phenotypes, and you know, the fact that it works about as well between siblings as between random people in the population is a strong indicator that you're not picking up some weird non-causal effects or some weird correlations with environmental effects. You really are picking up the genetic causes.
So Steve, imagine five years from now, or now, could you rank, let's say, 10 embryos that were of the same batch, same parents, from likely tallest to likely shortest?
Yes. Now, again, we don't offer this, but because there's a big ethical debate about whether one should rank embryos on a trait like height. Certainly, we are approached by parents who want this, but we don't offer it. But just as a scientific question, to answer your question scientifically, we can already do a pretty good job of ranking 10 embryos by height. And the cases that we call wrong would typically be a case where the brothers are actually pretty close in height. So in other words, like, maybe they only differ in height by 1 inch, and we mistakenly said the shorter one was going to be the taller one. That would be an error, but a very minor error, because we're really only making an error by 1 inch or something, right? Whereas if the taller brothers in the set of 10 are much taller than the shorter brothers at the other end, we would get that right. And so we can already kind of do that now for height, but it'll just get better. Okay. Now, this is, again, some needlessly complicated, geeky graphs, but let me just go through it quickly. What this shows is that if you look at increasing polygenic risk score, the probability that the person actually has the condition shoots up. Okay, so that's what's kind of being shown here. And what we did is we compared that for ordinary people, non-siblings, and we looked at specifically cases where one sib has the condition, and then we looked at whether the score would predict how likely the other sibling was to have the condition. So this simulates the situation where there's a family history. The whole family has an overall higher set of risk scores than the general population. And we ask, okay, but if you have a family where there's elevated risk in the family, but we managed to find an embryo which is low risk, even though it was produced in that family, it has a low risk score, does that embryo actually have a low risk of having the condition? And the answer is yes. So it turns out even conditional on having a sibling who is affected by the condition, we can still identify low risk embryos despite the fact there's some family prevalence. And so again, we're getting slightly into more things which can't be explained that easily to lay people, but this has a very specific application in IVF where you know there's a family history So there's an enhanced probability that among the embryos produced, more of them will be high-risk outliers. But we wanted to check whether a low-risk score among the siblings would still mean lower than average risk of getting the condition later in life. And it works. It basically works, basically about as well as it works in the ordinary population. So happy to answer any questions about that.
And in certain things, you could compute the probability of that. So, we're used to, in our very simple IVF world, we're used to sort of, 25, 50, and 100% risk. Whereas in this situation, you're dealing with things that would have a much, much lower probability, but it might just have popped up in one of the embryos.
Yes. Now the vertical axis on these graphs actually is the probability. So in other words, these are individuals who we would say there's a 70% chance they're going to have hypertension later in life. And these are people who have for example, a much lower probability than this is typical in the population, this red line. And these are people that are at way lower risk than what's typical in the population. So again, the results encompass people who are low risk outliers and high risk outliers. And it's something that we now show based on these results that we're pretty confident we could make that differentiation at the embryo level.
So based on this, would it not make a lot of sense in terms of building out the data sets to sequence as many old people around the world as we possibly can and get all of their information so that we can just work backwards from toward the end of life?
Absolutely, Jamie. The research value of the genotype of an individual, it's like, say it's going to cost you $100 per person to genotype. You get the most bang from your buck from people who are near the end of life. In fact, actually, the best thing would be if this sounds a little morbid, but if they just died, because then you get their whole medical history and you even know their lifespan. And you can use that to train, for example, a lifespan predictor. So yes, the older they are, actually, the more valuable, because if you have good medical records for them, the more valuable is their DNA.
And so would this also suggest that societies that really want to double down on the future of precision medicine should be working towards standardizing electronic health and life records so that these data sets can be maximally usable?
Yes, in fact, there's a country called Estonia, which despite being a former Soviet country, is kind of a Nordic country ethnically, and so they've developed all kinds of very forward-looking e-government, and they have an electronic health record that's very easy to access. And so they're among the leaders actually in genomics research now, because once they genotype a bunch of people, they can just immediately call up the data and start doing analyses. So that's the future we should all head for.
So Steve, what do you say to people who might say to you, yeah, but everything I've been reading in the magazines tells us that, well, if you have a Mediterranean diet, you're less likely to have such and such a disease. If you have a Japanese diet, you're not going to get hypertension and etc. So really, how powerful is this DNA when all you have to do is have the right diet?
Yeah, as I said at the beginning, we're not claiming that DNA is 100% determinative. So in other words, let's suppose that you're an individual that, according to the polygenic risk score, is at high risk for coronary artery disease. But suppose that maybe even you knew this when you were young, thanks to genomics. Imagine that you live a very healthy lifestyle, you exercise a lot, you keep your weight under control, you eat very sensibly. It could be that your actual risk is in the normal range, even though genetically you're super high risk. Maybe your environmental habits, your personal habits, like your diet and exercise, are pushing that risk back down to the normal range. So, we're not saying that you can do nothing. Obviously, there are non-genetic aspects to all of these risks which could be under your control. And so those are all important areas of study.
And of course, the converse of what I was saying, wouldn't it be maybe genomic predictions work will actually tell us that there's a lot more to the DNA than people believe when they were saying that the Japanese diet is what's important. It may be that the Japanese polymorphisms are important more than their diet.
Well, that's certainly possible. I mean, of course, it's also possible that a particular regimen that people are telling you is great doesn't really work that well, and their results are confounded. Actually, it was the genetics of the Sardinians or the genetics of the Japanese that actually led them to have low rates of heart disease or something. So we can't know that. Modern researchers now realize that if you want to establish a finding, say the effect of fish oil or a diet, you should also take into account the control for the genetics of the populations in your study.
Indeed. Thank you.
So let me go, see, was this our last slide? It might have been. Or it could be my computer just froze. I think that might be it. So, no, I went the wrong way. Let's see. Yeah, I think that's it. So that's the end of my slides. And now I'm happy to discuss anything you guys want to discuss. In fact, maybe I can get rid of this on my screen.
So I'm going to ask Jamie and you. If I was in charge of the National Health Service. Oh, sorry, I've got my voice coming back.
Oh, is that my fault?
Anyway, I'd be getting pretty excited that I actually might be able in time, given that we don't work with a normal 5-year political span, but you're actually looking to a nation with a 20, 25-year view, that we may be able to change general healthcare economics with this kind of information.
So everybody seems to be frozen except me. So I'll answer. And there also seems to be an echo. Should we wait to have this echo fixed, or should we keep charging forward?
So I heard you very clearly then, and in fact, I think my echo stopped.
Okay, good. So absolutely. I mean, I think that's the goal, is to move from generalized healthcare where people show up with a symptom, which is the most expensive way of treating, to precision healthcare based on individual biology, to the gold standard of predictive, because then the interventions are the least expensive and the highest value. And certainly one option is through empowered embryo selection, which is what genomic prediction is about. But even without that, if everybody knows what their risk factors are from early on in life, and you either make lifestyle changes in order to prevent bad outcomes, or test early where there are areas of higher risk, the economics of this, it would pay for itself conceivably. But that's the goal, is to build out those kind of models. And then there's a question of what is the role that the state plays when it's a national health service, or private industry plays in places like here in the United States, where it's more of a hybrid model. But certainly, conceivably, the costs of healthcare could go down dramatically if we shift from treating symptoms to preventing bad outcomes.
So
Steve?
Jamie, so just to pick you up on the US,
yeah,
the interim, of course, is insurance, isn't it? I mean, it's a massive player in the US. They may well be very clearly looking at this kind of thing.
Well, you would think so in a rational system, even a rational system based on private insurance. But the average American switches healthcare providers every 18 months. And so the healthcare provider doesn't necessarily have a long-term interest in the health and well-being of the individual. And because if you know, according to Steve's charts, that you have a much higher risk of hypertension, but it's 30, 40, 50 years from now, it would be, in an ideal world or in a single payer world, whether it's private or public, that single payer would rather make a small investment now versus a large investment later. But if it's a different payer, the goal is just to spend as little at each point along the life. And that's why these kinds of systems make most sense either in a world where there's a single payer, ideally a government single payer, or when individuals, whether they're parents or patients, are empowered and educated to think about their long-term health and well-being. Steve, are you back?
I'm back. Sorry if I was frozen. Yeah, that was well said, Jamie. I really think that in a system like the UK NHS, the incentives are properly aligned. If they can figure out how to do treatment better, they'll save money in the long run, and the long run is what matters to them. But unfortunately, the US doesn't have that set of incentives right now.
So could I ask another almost off the chart question? So I'm sitting here listening to this discussion, and if I'm an IVF player, I know all about genomic prediction because it's so cutting edge, it's so exciting. But what happens if I know that I'm going to have a baby, I'm pregnant or whatever, naturally, or I've gone through IVF but I haven't had the chance to do? Could I come to you and say, well, actually, soon after my baby's born, can you do the test for me?
Yes, absolutely. I mean, the main issue is just regulatory. Is once it is a being that has been born, are there different regulations about our ability to do a genetic test and return the results to that individual? And it varies from place to place. The way that embryo genetic testing is regulated and the way that adults or already living people are medical care for them is regulated. It just differs. But if that is not an issue, then for sure we can do it. And in fact, we as part of our process, which is kind of unique, because we're only using the DNA from a few cells, as you know, to do the genotyping of the embryo, we do something called error correction. We take saliva samples from the mother and the father, we genotype them, and we use that to error correct any noise that has crept in the process for the embryo. And what we've shown in some papers that we published is that the accuracy of the embryo genotype is higher than what you would get, say, from 23andMe or Ancestry.com right now. So it's 99.7 or so accurate. But along the way, we, as part of this error correction process, we get the mother and the father's genotype. So we can compute all of these risk scores for the mother and the father as part of our IVF process. And if we're allowed to report the results back to them, then we will report back to them, and the father may learn he's got to watch himself because he's got high risk for coronary artery disease, or the mother may learn she's high risk for breast cancer.
And I think that's if we can get past, if there's a regulatory hurdle and there's a cultural hurdle of just the way people think about life, but it just seems highly actionable that if people had this information and they just, you're at greater than average risk for type 2 diabetes, you should exercise and you should eat healthy, and you should start getting these tests that normally you would have later in life, earlier in life. I mean, I think that if the story can be simplified, it could be easily absorbable by people.
There's a certain irony there, isn't there, that there's going to be this huge debate, and it'll continue on whether we do this on embryos or not. But as soon as the baby's born, all parents, and then that baby, when it becomes an adult in its own right, will spend every moment it can thinking how it can make itself the healthiest.
It's true, and yet so many people are afraid of having their whole genome sequenced, afraid of getting information about future states that they may not want to know about. And so it seems from all of our perspective that it's a no-brainer. But I think for a lot of people, it's complicated. For a lot of people, there's this idea of fate, and this cuts against fate. And there's a fear, and Steve said it right, that this is not an absolute predictor of people's lives, but there are lots of people who don't want to know that they have a 25% greater than average risk for a certain type of cancer. And so I think that it's that all of this, from a scientific perspective, has one set of meanings. From a broader societal and cultural perspective, it's more complicated.
Very interesting. So as a futurist, do you ever think that government would insist that they have that knowledge? For the
I think, yeah,
I think
economics. I think it will make sense. I think that at some point in the not too distant future, I mean, right now, most governments mandate that newborns, for example, get a heel prick blood test. And so I think that it will make sense that every newborn will have their whole genome sequenced, which will be one of the foundations of their electronic health record. It's just to do that, it's not just a scientific question, it's a broader societal question. And that's why these kinds of conversations are so important.
One thing I'll just add is that the cost of genotyping is so low now that it really is among the least expensive medical tests that you can do. So if you give a newborn baby a blood test, the cost of that is pretty similar to, at minimum, it's close to basically doing the genome. And so, eventually it'll happen. It's really it's just a matter of the mental state of society that will determine when it happens.
Steve, have you got anybody on staff or colleagues that you're working with that can compute, just take one of those polygenic risks, that can compute the potential financial rewards, the financial savings in one particular nation? And we can do this in some diseases, we can do this in some chromosome conditions, but in terms of the polygenic risk score, is somebody working on what the potential financial advantages could be to a nation where we know, for example, that managing your lifestyle minimizes your risk of having the severe disease?
So I have some of that in my.
Sorry,
Steve. I have some of that in my book, Hacking Darwin. But there are a number of people who are doing those kinds of calculations, and if you do it, I mean, the savings are huge. But it's a societal shift. Steve?
Yes, the specific calculation that we did, and we published this in one of our papers, is for breast cancer, and we were able to do this calculation because we have the BRCA example. And so we know how much money the health system saves if they detect the breast cancer, or say 10 years early, through early screening that they would allocate to BRCA carriers. So using those inputs, what we showed is, roughly speaking, if you were to genotype all the women in the population, the cost savings from catching breast cancer early in the high-risk few percent, I think just 1% of women who are at the highest risk, would pay for all the rest.
Oh.
So if you believed my numbers and you just said, we're going to order the free genotyping of all the women in the NHS system, but then we'll carefully watch the top 1% in risk women, just like we watch the BRCA carriers, and we'll get to treat them early, and we'll save tens of thousands or maybe 100,000 pounds by catching them early. That will pay for the whole program, and that's what the numbers said to us.
And that's why doing these kinds of testing in the countries with rational national health systems makes so much sense. And certainly from a US perspective, the hope is that these ideas can be proven in places like the UK and Israel, and because it would be unfortunate, given how powerful these technologies are, if it's just a small number of people paying for themselves who have access to them here.
Oh, indeed. Indeed. I mean, I presume you're coming up with many of the arguments against this, like we did in the early IVF days, about it's only for the rich and etc. But yeah, those arguments eventually will never hold up.
Yes, and I think that these procedures are, as I was saying, already very inexpensive. If the health system can afford to just test your cholesterol level, it can also afford to genotype you, actually. And so it doesn't have to stay in the domain of the wealthy. It can easily, like I said, the breast cancer example, the national government can save money by just genotyping all the women, and then it isn't just in the hands of the rich, immediately so.
Steve, I don't always take up much more of your time, but I was just interested to know what your future goals are here in terms of, because we know there's a vast number of polygenic, or diseases are polygenic in nature. What would be your 5-year plan? Would it be to hone what you have or to expand the number of conditions?
The great thing about things which are informatical, computer driven, is that once you've written the code, once you understand how to analyze it, it's very inexpensive to do. And so we don't feel constrained to limit ourselves to any of those specific one things that you mentioned. So we intend to expand the set of diseases for which we can do prediction, improve the quality of prediction for each of those predictors, and push to not just have this available to families in IVF, but also work to get it into health systems so that an individual can know when they're young that, hey, they've got to watch what they eat because they could be diabetic later. So I really think all those things are technologically possible, almost inevitable in the next 5 or 10 years. And the question of the limiting throttling factor is society. Can society, and that's where Jamie comes in, and that's where you come in as thought leaders. Because if Jamie's book were read by President Trump and President Biden or whoever it is, it would, I think, further this whole thing, more than anything actually that technologists like me can do, because the technology is actually already there. And it's really just a light bulb has to go off in enough people's heads for this to move forward.